Okay, as we said in class, cellular respiration is something that occurs in all organisms, prokaryote, eukaryote. It happens in plants as well as animals. I know that in middle school, elementary school, a lot of times we were taught that plants put out oxygen and take in carbon dioxide, and animals put out carbon dioxide and take in oxygen. That's true. But plants also take in oxygen, and they use that oxygen to break down their foods. The difference is that plants make their own food, where animals take that food from plants. As we look at the process of cellular respiration, there's 3 steps. There's glycolysis, which is the first step in cellular respiration. There's a citric acid cycle, which is the second step in cellular respiration. And the third step is oxidative phosphorylation. We'll understand the terms a little bit better as we hit each one of these 3 steps. Understand that this is only when it's aerobic respiration. The first step does not require aerobic respiration, does not require first step of cellular respiration does not require oxygen. So it has really, and it occurs in both aerobic and anaerobic respiration. So it doesn't demand oxygen. This part here happens whether there's oxygen or not. These 2 require oxygen. But let's talk about glycolysis. Glycolysis is when we take this glucose molecule and split it up. And remember, glucose is C6H12O6. So because of C6H12O6, we know we have a 6-carbon long molecule. And as such, if we have 1, 2, 3, 4, 5, 6 carbons long, all of them attached one to the other. And remember, as we discussed in class, usually you'll find it as a hexose. It'll be a ring structure with the oxygen being here and the carbon sticking up. So that's kind of what we're expecting to see when we're talking about glucose. It's a straight chain in some circumstances and under some certain conditions, and usually we find it in a cyclic form, and it'll either be in 1 or 2 primary conformations. But irrelevant, the point is it's 6 carbons long, right? And glycolysis is just going to take these 6 carbons and in a certain phase of glycolysis, it's going to split it up into 2, 3 carbon molecules. There's many steps to it. In the next slide, I'll show you how complicated it is, and I don't want you to learn all the complicated steps. But what I do want you to learn, what you need to learn for the exam and for the end of course exam, is you need to understand that this glucose molecule is going to produce energy, and the energy that we're looking at is ATP. And we talked a little bit about ATP, and I'll take a moment later on in the video to make sure we understand what ATP is. But in the first phase of glycolysis, we take glucose and we add 2 phosphates to the molecule glucose, one on one side, one here and one here. And that preps that molecule to be able to be split. So the process of splitting it takes a little bit of effort. We call this the energy investment phase because we've taken energy in the form of ATP and put 2 of them into the molecule. So we've invested in that molecule 2 ATPs. The energy payoff phase, on the other hand, is where we get those ATPs back. And as we take that energy, as that molecule split it up into 2 pyruvates, we end up with 4 ATPs and 4 inorganic phosphates, or 4 phosphates, that's oxygen, 4 phosphorus with 4 oxygens, and we had 4 ATPs is what we form, with 4 ADPs, 4 phosphates, and we produce 4 ATPs. So we used 2, we made 4, so we have a net of 2. We have 2 net, so -2 plus 4 equals plus 2. That's your ATP. That's how much you formed. So you make a total of 4, but Realistically, you used 2, so you made a net of 2. Now, the other thing that we need is we need this NADH, because we need to carry these electrons that we're getting from this glucose molecule. We need to take these electrons and some hydrogen ions, and we need to take them and transport them to the electron transport chain. Otherwise, the system's really not going to work, and we're not going to get enough energy for our cells to live. So we need this NADH as an electron carrier. Don't forget that. We need this as a form of ATP. That does help, but that's not really the purpose of glycolysis. It's not the only purpose, rather. There's still a lot of energy in these 2 pyruvate molecules. And what are these 2 pyruvate molecules? Those 2 pyruvate molecules are those oxygen, that C6, broken up into 2 3 carbon molecules. And, we have 2 of them, and they're pyruvate. What exactly is pyruvate? It's a molecule. I'm not going to get into its shape. We can look up a picture of it and show it to you. It wouldn't do much good. Just know that we're taking this glucose molecule, we're splitting it. That's why we call this glycolysis. We're breaking up glucose, right? So we're splitting into two pyruvates, one here and one here. And we're also getting some NADH, which is important. And we did get some ATP, which is nothing to sneeze at. And so those are the 3 products from glycolysis. So we can see here that a summary, the net, right? Glucose, we're making 2 pyruvates and 2 waters. And here's the water being formed here. We're making 4 ATPs plus, but we used 2. So 4 minus 2 is 2 ATPs. And we use 2 NAD pluses. We have 4 electrons that came off the glucose and 4 hydrogens. And what we made is 2 NADHs plus 2 H pluses. And so this is our net for that particular reaction. Okay, so now let's go ahead and take a closer look and slow it down a little bit and see what we're talking about with all these major steps. We have glucose coming in. This glycolysis occurs here. And then once we get pyruvate, we oxidize it. We get in a Krebs cycle, and then we go to the electron transport chain. We'll look into what each of these steps are. But right now we're focusing on this glycolysis. So as we're in glycolysis, the first step is you have to have this 6 carbon sugar molecule, right? We need to break it down to these 3 carbon pyruvate molecules. And the first phase of that, as we said earlier in the previous slide, rewind this if you don't remember, is we have to invest. We have to invest some ATP. All right, so that's the first thing we have to do. And so you'll see here in 2 places, once here and then once again here, we add phosphate so that we can get on each side of this glucose, we get 2 phosphates. Now you notice that it changed to fructose. That's because there's an isomerase here that changes glucose to fructose. That's not really important. Each of these steps requires an enzyme, and when you're in college, you'll memorize the enzymes. You'll memorize the controlling step. You'll memorize the allosteric inhibitors and all the control steps and all the nice, beautiful details that you need to know. And it really is beautiful and interesting. But for now, and let's keep it simple, you don't have to know the names of these molecules, and you don't have to know the names of the enzymes. But what you should know, and you definitely need to know if you want to do well on the exams, is that you take 2 ATPs and you're putting them into this phase of the process called glycolysis. At the next step is, at this step right here, this is cleavage, right? And so at this stage, what we're doing is we're cleaving that molecule into 2 parts. And that's what this did. That's what this step did. It took this 6 carbon sugar and it broke it down into 3 glyceraldehyde 3 phosphate. So you don't need to know glyceraldehyde 3 phosphate. It's an important molecule. If this were an AP class, I would tell you need to know it. But I don't think you need to know it for your course exam, and you certainly don't need to know it for my test. I'm going to reference it, though, to that glycerol molecule that we used when we looked at triglycerides, right? So when you see these three carbons, glyceraldehyde, 3-phosphate, I recall the triglyceride discussion we had, where we talked about how we made phospholipids, where you took one of the carbons and you put a phosphate on it, and the other two had fatty acids, and triglycerides having 3 fatty acids. We discussed that. So they're all these molecules are related. We reuse them. I just want to point that out, that we didn't waste our time discussing the molecules of cleavage or ATP, or rather to make phospholipids, and neither do we waste our time, hopefully, in remembering what phosphates look like and what do lipids look like and what are sugars. So here are our biomolecules, and now we're using them. In any case, you invested ATP here, 2 of them. Here's cleavage. We cleaved it. We broke it up. Now we have 2 of these, and you do the same process, and there's in each of these, for each one of these G3Ps, you're gonna run them through each of these enzymes, and here's what you get is your payout phase, right? So out here is when you get paid back, and your payout is pretty cool because you invested 2 ATPs, and you're gonna get 4 out, and we saw that in the summary before. And you get 4 out here, you get 1 here, 1 here, 1 here, 1 here. That's 4. So 4 minus 2, the 2 that you invested, and you end up with a total payout of 2 ATPs. But you don't just get 2 ATPs. You get the bonus of, and it really is a big bonus, is this NADHs. And you get 2 pyruvates. And these 2 pyruvates are the key to what happens next. Because assuming you have oxygen, these pyruvates are going to go into the Calvin cycle. If you don't have oxygen, they go into fermentation, and we'll look and see what that happens during fermentation in just a second. All right. So let's take a minute and look at what happens when we don't have oxygen. So here you see that if there's oxygen, it's going, the pyruvates that we just talked about making, and that's this glycolysis stuff here, it goes straight in here, and if it has oxygen, it does the next steps of aerobic respiration, right? Cellular respiration. But if there's no oxygen, if you're running really fast, if you're a bacteria and it's in a place where there's no oxygen, if you're a yeast and you're in a place that's no oxygen, you need to be able to keep alive. And so you can take glucose and you can break it down. You can make enough ATP, especially if you're a bacteria, to stay alive. Your problem is that you're going to run out of NAD pluses because you're going to keep turning that into NADH every time you go down this path. And so you have to remake this NAD plus in order to keep this chain going, because it's like a game of hot potatoes, right? So you have to keep passing it. And if you have 100 hot potatoes and there's 30 of you, and you're not allowed to drop them, right? Because if you drop them, you lose. And you keep passing those hot potatoes down from person to person. You can only get 30 of them down the chain before finally that last person is going to start piling up hot potatoes. And at some point, she's going to drop them and the game's over. So this is what's happening here. These things keep being passed down one to the other. They keep getting used up. And at some point, you're going to run out of space. You're going to run out of NAD. You're going to run out of NAD8, NAD pluses. You're gonna run out of ADPs, because you're going to keep making these ATPs. We probably won't run out of ADPs, but you're going to run out of materials, specifically NAD pluses, when this glycolysis is going to stop. So you have to find something to remake this NAD plus, and the way you do it is you take this pyruvate and you run it through fermentation. This is fermentation. That's another chemical reaction. If you're a yeast or a bacteria, you're going to take this acetaldehyde, which is, you're going to make some CO2 right away with the pyruvate. You're going to take the 2 oxygens, you're going to take this acetaldehyde, and you're going to turn it into ethanol. All right. By doing that, you're using up NADH. And so NADH gets turned into NAD plus, which lets the NAD plus come back and take that electron and that hydrogen and keep the cycle going. As long as electrons keep getting taken, the game of hot potatoes can go on, or the chemical reactions can keep going on. Once you run out of electrons, once you run out of NAD pluses, the electrons can't be taken anymore, and the chemical reaction won't work. So here is where you transfer that electron. You transfer it to acetaldehyde, you make ethanol. Eventually, the yeast die from the ethanol, but that's how we ferment alcohol for drinks as humans, and that's how we make alcohol for sanitation purposes, etc. So ethanol is an important substance that we make using yeast, and yeast is also made to rise bread, but that's another discussion. Remember, yeast are fungi. But so, and we can also, some bacteria will also do ethanol fermentation instead of lactic acid fermentation. Now, humans and other animals, what happens is that in humans, this pyruvate is taken as whole, as a 3-carbon. Notice that this loses a carbon here, so you're producing carbon dioxide. This is why yeast make bread rise, because they produce carbon dioxide in anaerobic respiration. We don't produce carbon dioxide in anaerobic respiration. We take and we produce lactate or lactic acid. Lactate is the salt of lactic acid. So these 3 carbons from pyruvate get turned into lactic acid or lactate in our muscles. This is a problem if it builds up, if a lot of lactic acid builds up, because it could actually become deadly. So we can't go too long without with building up this lactic acid. But what's interesting is that we're able to make enough ATP to keep going for a short period of time without oxygen. And that's why we do fermentation, to keep being able to keep going when those periods of lacking oxygen. We can only make a small amount of ATP, but we make enough to keep going. So the next step in the cellular respiration is, after we finished taking that glucose, going through glycolysis, assuming we have oxygen, we're not doing that fermentation business. We have that glycolysis, we have made pyruvate. We take that pyruvate, we put it in the mitochondria. And in the intermembrane space, before it gets to the Krebs cycle or citric acid cycle, however you want to call it, remember they can be called either. You'll see it in the textbook in either way, as the Krebs cycle or the citric acid cycle. So again, from glycolysis, we produce 2 pyruvates, 2 3-carbon sugars, molecules. We produced 2 net ATPs. We produce some NADH. And these pyruvates enter here into the mitochondria when there's oxygen. And we'll see why oxygen is necessary in just a minute. And here, as pyruvate oxidation goes on, pyruvate is taken, 1 CO2 is removed, and acetyl-CoA is added to the 2 remaining carbons. If you have 3 carbons in a row, and you take 1 away, then you're left with 2 carbons, and these 2 carbons are attached to an acetyl-CoA. And acetyl-CoA. All right. And that's what allows it to enter the Krebs cycle or citric acid cycle. So taking those 2 leftover carbons and adding acetyl-CoA allows it to go into the citric acid cycle. We'll talk about that in just a second. And of course, one of the key points here is right here, before you even get to the citric acid cycle, you've already made a carbon dioxide. This is what gets exhaled. Where did that carbon come from? It came from glucose. It's the pyruvate the carbon from the pyruvate is what went into making CO2. But where'd the carbon in pyruvate come from? Glucose. So it got broken out and away it goes. That's only 1. Pyruvate has 3 of them. Where'd the other 2? What happens to the other 2? You lose them here. 2 of these end up leaving. So you end up with 2 CO2s leaving from the Krebs cycle, and 1 here and 1 here. And those 2 CO2s plus this CO2 up here equals 3. So there's your 3 carbons every time the wheel turns. You take out 1, 2, 3 carbons. Okay? And that's for every pyruvate, because every pyruvate only has 3 carbons. That's where your CO2s go. That's what you exhale. Now you have 2 of these, so you end up exhaling 6 carbons. And how many carbons did you start with in glucose? I hope you said 6. So 6 is the answer. So 1, 2, 3, and we're ready to go. So let's talk about this citric acid cycle or Krebs cycle a little bit more, and then ask about the electron transport chain. I think it's important to remember to point out that this happens in both animal cells and plant cells. A lot of people think that this happens only in animal cells. That's not true. This happens. Plant cells also have mitochondria. There they are. Okay, and these mitochondria, remember, through endosymbiosis, we think that mitochondria were first taken in as a relatively symbiotic host, really close host inside itself, and then later plants split off when they took chloroplasts. So that's a really important concept to remember. It's also important to understand that the carbon that we exhale is the food that we ate, right? So that's food that we've digested, and we've squeezed out the energy from it, and those 3 carbons are leaving our system in the form of carbon dioxide. You'll also notice that we produce ATP throughout here, and we produce NADHs. Remember, NADHs and FADH2 here are all going to the electron transport chain, and we'll look at that in a minute. Here you see GTP. Don't get confused. It's energetically equivalent to ATP. So you're producing 1 GTP is like producing 1 ATP. Don't worry about it. You remember what we talked about, these nucleotides. They're monomers of nucleic acid polymers, right? And if you have a G here, GTP, it's the same as having an ATP, except your base is different, right? Instead of an A, you have a G. You could have CTP. You have TTP or UTP. Right? It really doesn't matter. But in this case, you produce a GTP, and but energetically, it's the same as an ATP, or just as good. So these molecules leave in through the bloodstream. They get exhaled out. These go to the electron transport chain. You see it here. All these NADHs and FADH2s all go to the electron transport chain. These are all electron carriers. They all transport electrons. And now we'll go ahead and look at what exactly the electron transport chain is. I guess I should mention, though, before we go on, and I did in class already, that acetyl-CoA coenzyme A is a helper to enzymes. Each of these steps, as I said, have enzymes. If we take a look at how complicated it is, it gets really complicated. And so there's a lot of details here that we could just spend a good couple months on trying to figure out which enzyme does what. There's enzymes, all these ASEs are all enzymes. There are steps, there's changes, there are bond formations, and there's bending, and there's bonds made within themselves, intermediate groups. I mean, there's just all kinds of things that happen through the process, the chemical reactions that happen, that we've mapped it out pretty well. And we know what enzymes do the process, help the process along. We know where carbon dioxide leaves, where ATP, GTP enter and exit, NADH exit and enters, or NADH exits, NAD exits, enters. We know where citric acid comes out, succinyl, succinate, fumarate, all the different intermediates in the citric acid cycle. Now, citrate is one of the components. Do we really need to memorize that? Of course we don't. I don't want you to know this. Absolutely do not want you to know this. Well, I mean, if you know it, that's great, but that's not really what we're here to focus on. I just wanted you to see how complicated this is, and the fact that all these blues are enzymes, how many enzymes are involved in this one piece of cellular respiration. It's very complicated. The summary is all you need to know for your exam and for the end of course exam, but just know that the citric acid cycle, or the Krebs cycle, right, is a series of chemical reactions that are mediated by enzymes. And these enzymes have helpers, and these helpers are known as coenzymes, vitamins or minerals, etc. And this coenzyme helps this process go on because this acetyl-CoA, this CoA binds to the acetyl group and allows it to enter into the cycle. Once the molecule enters, the CoA leaves, and the group then gets transformed, or gets chemical reactions change that molecule into different intermediates until finally the original carbons that came in from the sugar leave as carbon dioxide. So when we're dealing with the mitochondria and we're looking at these NADHs all coming into the electron transport chain from glycolysis and from the Krebs cycle, and CO2 leaving here at this first pyruvate oxidation, and again from the Krebs cycle, 2 CO2 is leaving from down here. I know it only shows 1, but there's 2 leaving. We know that all the molecules of all the carbon from the glucose is all gone by the time we get to the electron transport chain. What we have left is the electrons and some of the hydrogen ions that came with the glucose still around by the time we get to the electron transport chain. So, again, this is happening in both plants and animals, so let's keep that in mind. And we have this enzyme, and this is a membrane. This was really great about membranes, right? And notice where the electron transport chain is happening, all right? So this is actually upside down here. So if you can imagine that if this were reversed, if we could just take this and just reverse it, all right? So it's upside down. I know you can't read it, but you don't have to read it. That's not the point here. So if we reverse it and we look at the membranes the way they are, then we see that this membrane here, right, is represented by this membrane here, right? So this ATP coming out here, making ATP, that's what's coming out here, right? So ATP and water, right? So all the hydrogen ions are being pumped, and they're being pumped up into this end, the opposite of this ATP. So it's this space here that gets filled with hydrogen ions. Let's take that and move it back around again. All right, so when we're talking about where are these hydrogen ions congregating, where are we making this high concentration of hydrogen ions, we're putting them all here. So, and we're making ATP by allowing them to run down the chemical osmotic gradient. But the key is that we're talking about hydrogen ions. So remember your pH. Remember that pH is equal to the negative log of the hydrogen ion concentration, right? And because it's a negative log, then this number here, pH, is inversely related to the concentration of hydrogen ions. So as you see hydrogen ions concentration increase, the pH becomes smaller. So that explains why a pH of 1 has more hydrogen ions in the solution than a pH of 11. Because the larger the number, because of this negative, the smaller the concentration was originally of hydrogen ions. So that being said, let's try to look at this electron transport chain a little more directly. And so we have this NADH and this FADH2. Where did this stuff come from? Where did this molecule here and this molecule here, where did this come from? They came from glycolysis and again from the citric acid cycle. So we got this NADH here, NADH here, NADH here, and FADH2 here. All of these came down to the electron transport chain, and this is where they end up helping. I want to point out something. We studied these membranes pretty well. You know that these are phospholipids. You know these are proteins embedded in the phospholipid bilayer. And you know what we can do when we create a high and a low. We know that once you get a high up here of high hydrogen ion concentration, right, a low pH, but a high hydrogen ion concentration, the hydrogen ions are going to go from high to low, right? So this is your high here, hydrogen ion concentration, and here is your low, all right? So the movement of ions is going to be from high hydrogen ion concentration up here to low down here, and it's going to happen that ion is these ions can only flow through this ATP synthase. So make sure you know the name of this as well, ATP, right? Synthase. He's running out of room. So ATP synthase then it's key to understanding what it's key to understanding how we make these ATPs, because this is what makes it right there, this molecule, this protein. So ATP synthase is allowing this high energy, high hydrogen ion concentration here to move. As it moves through the protein, it turns the protein. That turning opens up a space that the ADP can bind to. Look at the ADP 2 phosphates. That's those little yellow ones, and plus a phosphate together, they're going to make ATP 3 phosphates bonded on the ATP. That last one being very high energy. That's a little lightning bolt that's holding those two together. All because you're going from high to low. And that's great. And, I guess the question comes in, though, why do you need the electron transport? Why do you need the electrons at all if this is happening? And the second question that comes to mind as I look at this is, hey, wait a minute. What happens when all these hydrogens flow through this protein? Well, you go to an isotonic solution. You're no longer going to have any flow, right? You're going to have movement. It's going to go up and down just a little bit at a time, but it's not going to be in one direction. It's going to be in both directions, and it's not going to allow you to drive this consistent production of ATP. So you're going to run out of ATP and you're going to die. So we have to keep this high and we have to keep this low, right? How do we do that? How do we keep? We need a pump. We need an active transport system, something that uses energy to push, not allow things to go from high to low. That's too easy, but to allow things to go from low to high. That we need that pump. We need to be able to move these hydrogen ions down here against the hydrogen ion concentration gradient and go from low to high. You need energy to do that. That's what these electrons are for. That's why we need these electrons. They're a form of energy. And as we take these electrons and we put them up here and they transfer, what happens is that these are negative. What are they going to attract? What do opposites do? They attract these little these little golden things, these electrons. They're negative, and they're going to pull the hydrogens in. But unfortunately, this is a one-way trap, like a Roach Motel. It's going to allow them to come in, but it won't let them come out. So the electrons move down the chain here, and with each move, they bring in some hydrogens. All right, so the NADHs bring in some hydrogen as they go. Then the FADH2s bring some in. And all these electrons start to pull the hydrogens back across the membrane from low to high. All right, and that's great, but these are all carriers. All these are holding electrons. What happens when you get to the end and all these holders are full? Well, something has to take these electrons away. What could we possibly use to take the electrons away? We use oxygen. So we take oxygen gas that we have, that we breathe in, right? Half an oxygen molecule. We add a couple hydrogen ions plus 2 electrons, and what happens is that they go away in the form of water. You urinate it out, or it goes into your bloodstream. What's cool is you're getting rid of more hydrogen, so this even becomes lower. And this becomes water, and water is a solvent, and it can go through the membrane any way it wants. And as you build up water, that just goes away. And so oxygen ends up taking is the final electron acceptor. Oxygen is the final electron acceptor. And as it takes that final electron, it goes and makes water, and it takes those electrons and those hydrogens away, and everybody's happy. So make sure you understand that that's what oxygen is doing. And we're doing this whole electron transport chain is just to make a high concentration on one side, a low on the other. It's just so ATP synthase can have hydrogens run through it, make ATP. This ATP is used all over your body to do all the things that you need it to do. And this chain is taken away by oxygen. This is why you breathe. This is why you have to take a breath, because oxygen takes those electrons and it gets turned into water again. All right, so that's it. That's it for cellular respiration. I hope it's clear. I'll be sending you the PowerPoints as well with this. I covered it in class. This video is obviously not mandatory. I just wanted to help you out if some of you didn't get it the first time in class. I hope I was clear. If you need me to clarify something, let me know in class, or ask me to make another video that's slower or whatever else you need. There's plenty of videos out there that are faster, better prepared, and also some that are slower, some even rap. So you can find your own videos as well. All right? So good luck studying. Bye-bye.